I think where we're going to end up is at a place where we have instructions that are fairly generic, I'm hopeful, so that for a particular manufacturer's type of scope, they're going to always say apply air in this manner. What may change maybe is the pressure or maybe the duration. But I really am hopeful that we'll be able to have a general set of instructions so that it's not unique to every single model, every single type of device, because that'll just be so difficult.
Public health. It's on everyone's mind these days, from hand washing to emerging pathogens. Welcome to Transmission Control, an infection prevention podcast focused on your appetite for trailblazing thought, discussion, and innovations that will help you make informed decisions. Each episode, we speak with public health experts and safety champions from across the globe as they share their experiences, passion, and opinions. From investigative journalism to medical publications, we tackle real-world barriers to halting the spread of disease. Whether you are tuning in for education, inspiration, or to hear the stories that need to be told, thank you for joining us. And now, get ready to blast off with your weekly injection of insight on transmission control.
This week on Transmission Control, we speak with Nancy Robinson and Tamara Behm from STERIS Infection Prevention Technologies. Nancy has her Ph.D. in biochemistry from Case Western Reserve University, and for the last 23 years has worked for STERIS Corporation, developing liquid chemical sterilization and high-level disinfection endoscope reprocessing systems, vaporized hydrogen peroxide sterilizers, and more recently, investigating how to effectively dry the internal channels of flexible endoscopes prior to storage or sterilization. Before joining STERIS, she was a captain in the Army performing research at the United States Army Medical Research Institute of Infectious Diseases at Fort Detrick in Frederick, Maryland, as well as an academic researcher at Case Western Reserve University in Cleveland, Ohio. And Tamara Behm, clinical education specialist, holds a master's in nursing administration from Fort Hays State University. She is currently a clinical education specialist with 15 years of vast clinical nursing leadership and healthcare experience. She has held various roles as a director of ICU nurse, director of infection prevention consultant, infection prevention mentor, college adjunct faculty, presenter, author, and mock surveyor. She is an expert in infection prevention and ensuring facilities are meeting CMS and other regulatory requirements, as well as decreasing infections through process improvement projects in acute care, ASCs, clinics, and SPDs. She is a member of APIC, SHEA, AORN, ISHM, SGNA, ASQ, AMI, and holds an additional certification through the AHA of ASHE Healthcare Physical Environment Worker certification. So I'm really excited to have them on the show today. And Larry, really, one of the reasons that we reached out to them is this study that was featured in the American Journal of Infection Control. And the article that was talking about the findings and the process of how the study was conducted was called Efficacy of Flexible Endoscope Drying Using Novel Endoscope Test Articles that Allow Direct Visualization of the Internal Channel Systems. And these enhanced visual inspection technologies are fairly new to the industry, especially for the folks that are working on the front line. And it gives them information that they didn't have before. But in this case, it was used in a study to look at drying and how effective it is. And with all of the recent updates to the multi-society guidelines, Larry, I feel like this is just part of the rationale or the conversation or debate that's going on around some of those changes. Do you have kind of a perspective on those guidelines and how this can be so important to understanding how we're moving forward?
Yeah, there are a number of issues in the guidelines, of course, that are very important and are comprehensive and protect patients. And an issue that the guidelines always focus on is cleaning. And I'm kind of curious how this novel endoscope model that Nancy came up with and her colleagues at STERIS, how that can be used for other applications, such as maybe allowing us to better evaluate cleaning, maybe allow us to better evaluate damage. We use the borescope typically, and there are some data that suggest it can preemptively detect damage, at least in the suction channel that's wide enough. But I think this model can give us a lot of insight into what's going on inside the endoscope that we never thought was possible, or that we knew was happening, but we weren't able to quantify. We were only kind of qualifying, saying, yes, we have wear and tear. Well, how much? So this might be able to give us a lot of insight into where there are still significant room for improvement.
Excellent. Well, I'm looking forward to this highly engaging discussion, and we're going to be right back after a short break with Nancy Robinson and Tamara Behm.
Brought to you by 17 Studios. Let's get into it. This is Transmission Control.
Joining us now is Nancy Robinson and Tamara Behm, and we're going to be talking about a study recently featured in the American Journal of Infection Control. And it's a conversation that Larry and I have actually been having, and I think the whole industry has, for quite some time around drying and the use of alcohol. And this conversation today is going to be really important. So Nancy, first off, I want to welcome you to the show and thank you for joining us.
Well, thank you, Justin. I'm really happy to be here.
And Tamara, thank you for coming on as well, and really appreciate you joining us.
Thank you, Justin. Appreciate it.
So why don't we start, Nancy, with the study and just a very general overview. I know we're going to talk about some surprising findings, and I didn't just find them in the summary. I found them throughout the article regarding the study, but could you just talk to us about how the study came about, what the purpose was, and then maybe we can talk about some of the findings?
Okay. So for me personally, I became very interested in drying because I work with vaporized hydrogen peroxide sterilizers. And you have to dry your devices before they go in the sterilizer. So a lot of devices, that's pretty easy to do. They don't have channels. You just dry the external portions. It's not so bad. But when you come to something like a flexible endoscope, that's tough. You can't see inside a flexible endoscope. How do you know it's dry? So I became very interested in that, and that just naturally evolved into drying after high-level disinfection or liquid chemical sterilization. So we became very interested in that and trying to understand how you would control it. What's important for that? So when we looked into the literature, we couldn't find a whole lot of information. And we saw there were some methods. So you could use a borescope, and the borescopes are limited in the channels they can go in. They're quite large, and a lot of the channels within a flexible GI scope are very small. So you can't get into them. So one of the things that we were concerned about, especially when we talk to users, is, well, if I use a borescope and I know my instrument channel is dry, I know the device is dry. And I was a little concerned about that. And then the other thing is there are moisture detection papers that can be used. But talking to Tamara, who's with our customers all the time, she says she doesn't see that being used very much as a drying verification. So we wanted to find a different way to look at it. And so we developed these very unique test articles where we just stripped everything away from a flexible endoscope. So we bought some damaged flexible endoscopes fairly cheaply. You can buy them on the marketplace. They no longer had good optics or something. And we had them stripped away by a device repair company. And what we were left with was all the connections from the external endoscopes, and we could see all the channels. Think of it like the guts of an endoscope. You removed all the optics, you removed all the articulation. There's just all of these clear channels running around connected to the ports that are accessible from the outside of the endoscopes. And we use these as our means of saying what happens when we apply air here, what happens where we apply air there. And we came up with some really interesting results.
You know, it's interesting. I've used to work for a company that was in the instrument repair business, and I used to love watching the faces of the end users when they could actually see all those components laid out on a table and just not knowing how intricate really the design and how complex the design really is. And you talked about a new technology, borescopes, to be able to look into those channels. And this is definitely a good application for that. And that technology doesn't just apply for inspecting, but it's new. So many people already don't have adoption of that technology to be able to go in and see what's inside of the scopes for themselves. So, it kind of connects that what we don't know and how do we then take that and make it more objective, which is why I really liked the study. I felt like we were getting more objective information. So one of the findings that I read, or at least one of the observations, was that the small diameter scopes, they're not necessarily accessible by borescopes today because of the size of the diameters. And there were some implications there. What were some of the other surprising findings from the study?
Well, the most surprising thing to us was we expected if we flushed the channels with alcohol, 70% alcohol, they would dry faster than if we didn't flush them, if we just rinsed with water. And we found the opposite, that when we flushed with alcohol, it took longer to remove and dry the alcohol than it did if we did not flush with alcohol. That's very surprising and unexpected, because you expect alcohol, if everyone's experience with alcohol is if you look at alcohol and you blow air across it, it'll evaporate faster than water. That's absolutely true. The difference is this is within a closed channel and you're not blowing air across it and trying to evaporate. What you're actually doing is blowing air to force the fluids out of the channels. And you can force water out quite easily. But the alcohol, it kind of stretches and coats, and it stays within the channels. So it takes longer to actually dry it.
And Nancy, did you find this for each of the endoscope channels, the suction channel as well, or just for the narrower air water that kind of merged from two channels into one?
Well, it's interesting, Larry, because our data was really, really messy, I like to call it. We had a lot of standard deviation. I like doing experiments where I have really tight standard deviation. Unfortunate thing is, because the amount of water or alcohol left behind was variable from time to time to time, our results were quite broad. So we would do the experiment again and we'd get a big difference in the time that it took to dry. But we felt it was important to look at what a real user would do instead of installing a known amount of fluid in there and trying to understand the drying. We wanted to get a more real world picture of what was going on. So we had a lot of overlap in our data. So we found that the air-water channel was one of the channels that did not have this real difference between the alcohol and the water, while we did see the difference in the auxiliary water channel as well as the suction instrument channel.
Very, very interesting. I found that finding remarkable, and I would like to talk to you a little bit shortly about the IFUs and about drying protocols and what the manufacturers say. And dots I had not connected on my own until I read your article, that the IFUs that we see in the endoscope operator manuals generally say dry the endoscope, or they will say flush it, but we don't get into the actual step-by-step particulars. We don't just say clean the endoscope. We say walk through these 20, 30, 50, 100 steps. And I had not appreciated that your work is suggesting that I was wrong, that maybe we do need to do 2, 3, 5 steps for the drying, not as much as for the cleaning. But when you were talking about the evaporation and the alcohol, I want to just tie a loose end for my own edification. When you said the alcohol didn't evaporate or was not dispelled from the channels as quickly, might that explain, do you think, why the alcohol might potentially have a disinfecting properties to it that you've talked about, or maybe even fixative properties? Is it because the alcohol is staying around longer, do you think?
Larry, I love your question because it's coming out in a new article that we are writing to look at what is going on with alcohol and how does it compare to water. And the answer that we've come up with is no, that's not what it is. Alcohol really, at least for the organism we use, which was Pseudomonas aeruginosa, it almost immediately inactivated the organism very, very effectively. So I don't think that it's hanging around doing it, but again, that's one organism, and we know there's a lot of organisms out there, and we know that alcohol is not a sterilant. Alcohol is a disinfectant, and it has various efficacy for different organisms. So I don't want to say that alcohol is the end-all and be-all to stop outgrowth, but I think it can be a real nice, it's one of the pros for using it.
And you talked about that in your article, and you kind of segued into something that I really am curious of your thoughts on. And it had to do with, you discussed it in your article. We've talked about it now for probably a quarter of a century. It's in all of the guidelines, SGNA, ASGE, APEC, etc. Can you give us a little insight as to whether you think the alcohol might be drying the endoscope, and the drying would be eradicating, destroying that Pseudomonas you talked about in your upcoming study? Is it the intermediate level disinfecting properties of the alcohol that could be destroying the Pseudomonas? Can you give us a little insight into what the alcohol is doing? More facilitating drying, more microbactericidal properties? What are your thoughts on that?
What we were able to show by comparing water to alcohol is that the water did not have any sort of, if it was wet still, you had outgrowth. So we looked at suboptimal drying, optimal drying, and super optimal drying. So just to see if we got some more bang from doing more drying. And I'm going into our next paper, so you're getting a preview here. But what we found was that the alcohol was very effective, even if you didn't dry it afterwards. If you just flushed with alcohol and then you recovered, you would get a huge reduction in your recoverable bacteria compared to water. So there was definitely an effect. And drying, really, we didn't even see any effect of the drying. There was no greater effect by drying.
Excellent.
We would definitely want to hear about some recommendations that you might have as a result of the study. But I want to make sure that we have really talked about the way that the industry is today. And I think it's a great opportunity to bring Tamara into the conversation. So, Tamara, can you talk about the current standards? And then what I think everybody feels, there's so much opportunity in this area of medical devices, I think, for just, continuous improvement. Can you talk about those current standards and then maybe where there are some gaps in the real world in terms of the application?
Sure, Justin. When I visit our customers, it's often a question that comes about is questions related to drying, questions related to alcohol. And when you look at the standards, for example, AAMI ST91, it's very specific to mention that you need to flow air through all of your channels with 70 to 80% alcohol. And the majority of customers that actually are privileged enough to have an AER, their AER normally injects that alcohol as well as does a small purge of air. And many customers truly believe that is their drying that is advised by the manufacturer. And so they usually are fairly surprised when I explain to them that the AERs do not have clearance for that type of expectation, right? Because their purging is usually about a minute or so. And then when you also look at the standards, they mention consistently, multiple times, IFU, looking at the manufacturer's instructions for use and ensuring that you're following them. But what I often find is that facilities, number one, they don't have the means to dry. So they don't have instrument air or a filtered air of some type to dry their devices. On top of that, they may believe that the alcohol, like we spoke of, assists with the drying process. And they believe that drip drying it in their cabinet will actually dry it. A lot of customers don't understand their IFUs, where it actually will give some type of description of drying. And they truly believe that they're following them. But then when you pull the IFU on the model or the device, their eyes, pop open going, oh my gosh, I didn't realize that we weren't following the instructions for use. I've been doing it like this for years.
So Tamara and Nancy, do you have any insight for our listeners on something that I found was fascinating. And as I said earlier, it hadn't occurred to me, which it appears that if we're talking about a colonoscope or duodenoscope or a cystoscope or a bronchoscope, that the recommendation for drying is too generic. And I don't want to put words in your mouth, but it seems as if we simply say flush with alcohol followed by forced air drying. Your data are suggesting that we really need to customize the drying protocol, that the current protocols aren't validated per se. Again, I don't want to put words in your mouth, but I do want to hear what you say, and I know our listeners will be interested in knowing whether you're favoring more of a customization so that each of the drying protocols would use a similar paradigm to the cleaning protocol, which is maybe we need something specialized. If we have a bronchoscope with one suction channel, that drying instruction might be unique and separate from a drying instruction for a duodenoscope or for colonoscope with a auxiliary water jet channel. So can you share with our audience whether we really have been missing the boat a little bit and been too general in our drying IFUs and maybe should be more specific?
So I'll start off, Tamara. I think absolutely we need more validation of drying so that we're sure that we know that we're drying. We have good instructions. And I like how, Larry, you talk about cleaning. So I do liken where we're at in drying is what cleaning was like 20, 25 years ago, where we were debating how clean is clean. So they can ask that same question. How dry is dry? How do you know what's dry? So there's a lot of unanswered questions. I think where we're going to end up is at a place where we have instructions that are fairly generic, I'm hopeful, so that for a particular manufacturer's type of scope, they're going to always say apply air in this manner. What may change maybe is the pressure or maybe the duration. But I really am hopeful that we'll be able to have a general set of instructions so that it's not unique to every single model, every single type of device, because that'll just be so difficult. But when I think about cleaning, in general, cleaning is pretty much done the same. We flush certain channels, we brush certain channels, and I'm hopeful that something like that will be the ultimate outcome. But I do believe it will have to be validated, just like manufacturers validated cleaning instructions. I think it needs to be validated by the manufacturer. So now I'm going to turn it over to Tamara so she can give her opinion.
Yeah, that's right, Nancy. We definitely need to raise the awareness specifically with manufacturers that customers are actually unable to complete the drying process safely currently. And I believe that all of the literature that Nancy has brought up, all of the standards have recognized it. Other organizations have recognized it professionally, that it is a gap. So now we need definitely the manufacturers to bring this to the table and either give very strict instructions by scope and or providing also acknowledgement that either alcohol should be used or should not be used. It's important because ultimately we have to remember that this isn't just, for example, a GI area or a place you're having a bronchoscopy, but I also visit urology clinics across, all my states that I'm a part of. I visit ENT clinics, and we have to remember there's so many different types of scopes, and some of them are super delicate little pediatric scopes and things that we have to remember that the pressure, the time, and everything is very different depending on that size channel. And again, you run into the gap of supplies, knowledge, training.
Yeah, and really equipment. I mean, some of those clinic settings haven't made the same types of investments that you see in the more acute settings.
So are we in agreement then that it's interesting that when we think back 30 years, when the reports first came in ERCP, we found Pseudomonas outbreaks and fatalities, and we found in a number of cases that the facilities would implement this alcohol and this forced air drying, and in many cases that terminated the outbreak. Nothing else would do it. It's just interesting that 30 years later we're still talking about the conversation. But can we agree then that ideally we'd like to see operator manuals moving forward that would give us a specific validated drying time and a specific type of apparatus, whether it's the pistol, the air pistol, or whether it's the manufacturer's cleaning adapters or disinfecting adapters, that we'd like to have a particular mode of application, how we would apply the air, what the pressure of the air would be, and what the time of the drying would be. That would be getting into the specifics of the validated drying time. Do we agree that this is something that we think should be happening in the near future? Yes?
Yeah, Larry, absolutely. I think that this is something that we need to do, and I think that it has to be recognition not only by the medical device manufacturers, but the people that produce AERs, since the AERs do part of this. So we need to have all of us working together to solve this problem. We have to have the regulators, the customers, the AER and the reprocessing equipment manufacturers, and the flexible endoscope manufacturers all working together to solve this issue because it is critical. I like the fact that you brought up that 30 years ago, what we looked at was we're having these outbreaks, and yet you see in the literature when they started drying, when they started using alcohol, these things went away. So obviously we have data that says that it's effective and it's necessary, but we've gotten away from that somehow, and I'm not quite sure why.
That's an excellent point, Nancy. And two things. The first thing is maybe as an outcome of this podcast and our discussion today and listeners hearing our input and feedback and perspectives, that maybe the FDA would be able to convene maybe a guidance document to give some instructions and standardized kind of recipe for endoscope manufacturers and AER, the automated endoscope reprocessor manufacturers, so that the FDA could provide us with some guidance so we could move forward and kind of wrap this package up. And I had a second point, but did you want to interject, please?
I wanted to say something. Normally, I do a lot of work with the FDA because I am in research and development. And it's usually a collaborative effort between industry and FDA because FDA can look at things and guide us, but it is industry that develops the methods. It's industry that has the resources to do the research in order to figure out what should be done. So I don't think we can rely on FDA on its own to do this. I think it has to be done together.
Very good point, and I appreciate that very much. And I appreciated what you said earlier about we had been moving forward and maybe we have gone maybe walking things back a little bit, maybe not necessarily in the interest of public health. And that kind of catches my attention when you say that because I recently reviewed the 2011 Multi-Society GI Endoscope Reprocessing Guideline, and I compared the one from 2011 to an updated one from 2017. And then, as I'm sure you're familiar with, the new one came out in 2021. And where the 2011 and 2017 seem to emphasize the importance of drying in the alcohol rinse, the 2021 seemed, I don't know that I want to say walked it back, but did not seem so enthusiastic about that practice. And basically said to the effect that they identified one report about this, and so they didn't really take that much of a position or that strong a position on the use of alcohol and compressed air, and actually went as far as to say that there's no data that currently support or refute the use of alcohol flushes for the drying of endoscopes. Do you have a comment on that?
I do, Larry. So I just reviewed the 2021 guidelines as well for our company and kind of compared the 2016 guidelines. It was very interesting to see that the alcohol recommendations, and then in 2021, they did mention the fixative properties, that retention of organisms within the endoscopes could happen because of those fixative properties. But they also stated no strong data to refute alcohol flushes. And then on top of that, which even to me was a little more concerning, right, is we're talking about that customers don't necessarily have the tools to be successful. And the only tools that we know of that are even out there to know whether or not your scope is dry is either a borescope or some type of paper, right? I have not yet been into a GI or a scope area that understands that they have these types of options. And number one, if they don't even have air to dry their scopes, chances are they don't have a borescope. And so when I read the Multi-Society guidelines, it also said very specifically, no recommendation can be made for the routine use of borescopes. But if we don't have something to find out whether or not our items are dry, we have to use what resources we have out there. And it also said very specifically in the guidelines, it says additional larger and more rigorous studies of this new tool are needed. And they hit the nail on the head, right? So companies come in and we sell them a borescope and the customer doesn't know what to look for. They don't know the difference between water or simethicone, or they don't know the difference between a tear or biofilm. So along with that, for them to be successful, to know if there's water is to actually, be trained and have a good program for that as well. We have to arm our frontline workers with the knowledge and the support to dry safely and to be educated on how to do that as well.
That's a really good point, and I've seen that. I was kind of alluding to it in the earlier part of the conversation where there's just information that customers have never had before. And so a lot of times when I see them implement borescope technology, which I think getting some information is better than not having any, I think the adoption of this technology is positive movement. But to your point, there's all kinds of things that they find that they never knew, that they could never see before. And it even goes to the design of the scopes. Like I said, when you show people what it looks like, when you break it down into all these different parts and pieces. And so you've also talked about all the various different types of scopes. And so I have to think that you've put a decent amount of thought into like the impact of all those varying parameters in the way that the channels are designed or even the size of them. And when you talk about applying air pressure, you've already alluded to that. But how do all these different design factors and variables come together on maybe this customized sort of approach that we were talking about?
Justin, I'm going to take a stab at this. I don't think we know enough. I have found that drawing is so much more complex than I ever thought it would be. And when you apply air to a channel that branches and it goes in several directions, depending on where you put that air in, and if you're putting it in two openings to the same channel, sometimes the air at the same time, they fight each other. So it's very complex is what we're finding. And I don't have a really good answer to that. And that's disappointing. It really is disappointing.
But what we do know, as we said, from the past 30 years, that the drying really worked, and the alcohol, whether it was introducing a disinfecting property, as you talk about in your paper, or maybe as it was introducing, a evaporation, which maybe is not the case, but we found the 70% in the alcohol really helped. And so that left me a little confused with the guideline we were talking about earlier, Tamara, because they did talk about the protein, the fixative properties. And I think that's a valid point. But when I looked through the literature to try to find a case where an infection was linked to an increased risk of infection was linked to the use of alcohol, meaning that alcohol was directly linked to protein buildup that was then directly linked to an infection, I couldn't find it, yet I could find the cases where the alcohol was reducing the infections. Is this consistent with what you have found, what you conclude?
Yeah, Larry, I don't know of any real good articles. There's only one that's out there, and it wasn't really done for flexible endoscopes looking at fixative properties. Alcohol, it for sure will precipitate proteins. I'm a biochemist. That's what it does. But glutaraldehyde fixes proteins. It's a much better fixative or precipitant than is alcohol. So this is my own personal thoughts around the use of alcohol. One, it's extra steps, right? You have to then flush, especially if you're doing this manually. So we'll just start with a manual process. You have to flush those channels with alcohol.
Using a syringe.
Using a syringe, which is not easy to do, okay? So if instead we spent the time and the effort that we use for the alcohol flush doing an effective drying, we probably would come out in the same place, because I strongly believe that if we dry the channels, we will stop any outgrowth if you had either an ineffective disinfection process.
I agree.
Or you had a biofilm established so you weren't effective with your disinfectant process, or if you accidentally introduced some type of waterborne organism, or we just had environmental contamination. I feel strongly that if we have an effective drying, we don't need alcohol. Nancy's opinion.
So I have my opinion too, from a clinical perspective and an infection prevention perspective. I think that we are leaning towards many different avenues in the infection prevention world. Again, it's no longer control, it's prevention. That facilities definitely can look into doing some type of a risk assessment of their processing and investigate whether or not their devices are dry or not. We have guidance, very minimal guidance from our manufacturers on how to dry, but that doesn't mean we can't take that step as practitioners to go in and look at our scopes very clearly and see what we need to do to dry them. I think we have, the means. We have definitely a lot of wonderful literature. We have Nancy's paper, and we have a lot of guidelines that say they need to be dry. So if we take our scopes and we go in and do a risk assessment with a multidisciplinary team to decide by the model of scope, go in and look at them and see what do we need to do to dry. Because I think, like Nancy said, we have to all work together.
Yeah. And what you just talked about too was really just about process validation. And everybody should be doing process validation. So I got to tell you, this was an excellent conversation, and you both made some really good points. And I think it's important, for everybody out there to see sort of where the opportunities are. But I also like how you just told that anybody who's listening who works in this space, how they can be part of the solution. And that's so critical. So I want to thank you both for coming on. And I also want to give you an opportunity for some final thoughts. So Nancy, we'll start with you.
Okay. So my final thought is I want to be very careful when you read our article. We are not trying to define how to effectively dry. That was not the point. The point was to raise awareness of how that these parameters, where the air goes in, what pressure you use, what fluid is left in there, affects the ability to dry. And we were not trying to come up with a effective drying procedure because we felt that for doing it for one scope, people would apply that to all scopes. And I think that's dangerous. So I want to be very careful if you read the limitations or what we were trying to achieve in that article. We did not want people to walk away and say, now I know how to dry a scope. So I just want to make sure I make that clear. And the last thought is the users do not have the tools they need to verify dry. We need something, the industry needs to find a way to put in the hands of users a simple, relatively easy tests that will tell them whether their scope is dry or not. We do have some methodologies with the papers, the drying papers, and applying air to the channels using the drying papers that'll give you an idea. But it's not the end all or be all. So there's things that need to be developed for the future.
Lots of work to do. Tamara, any final thoughts from you?
Final thoughts are I really appreciate you guys for bringing this to a broader view of people and making them aware, because Nancy and I have just said this, and I'm super passionate about the fact that we need to make these procedures as safe as we can for patients. And the best way to do that is to just band together as a group and a team, and whether it be APIC and SGNA and the Multi-Society and AMI and us, we need to come together to support the people that are doing this work and make sure that they're supported with the tools, the equipment, the knowledge, and the science to ensure that they're processing and drying their scopes safely.
Excellent. Well, thank you both for coming on today. Really appreciate the conversation. That was Nancy Robinson and Tamara Behm talking about scope drying and I'd say, just the involvement or the role that alcohol has had in the past, and then some things from the article around the study featured in the American Journal of Infection Control that had some pretty surprising information, Larry. And I know that Tamara really outlined this pretty well, but that alcohol has played a role in scope drying this flush. and that the assumption has always been that it has helped with drying. And the study does not seem to indicate that that is the case.
No, I thought they did a really good job in teaching us about how little we know about something that we thought we knew everything about. That doesn't happen too often, and I was really impressed with that. They also discussed with us how kind of one shoe doesn't fit all feet, and that the issue with endoscope drying is we need to become more specific and understand better that for the model we're talking about, or the type, to simply have a shotgun approach to say alcohol flush and then dry is not doing the patient a service. It's a disservice. And what we really need to do is come up with more of a validated IFU, an instruction for use, that tells us how long should we be doing it, where should the air be applied, what should the pressure of the air be applied. And I hadn't thought about that. In all the years I've studied endoscope drying, they really shed light for me on something that I had not thought about, and I really appreciated them doing that.
Yeah, this is a very important interview, I think. And there is, as we said towards the end, more work still to be done. And I can see us having them back. And even after we got done with the interview, we talked about it and said, well, the next step is we got to talk about storage. So I would say we can expect a future episode around storage related to these scopes. But that's going to do it for this week's show. As a reminder, you can help support us by subscribing to Transmission Control on iTunes, Stitcher, Google Podcasts, and Spotify. We certainly appreciate a rating and review because your feedback is important to the show. And on behalf of Larry and myself, thank you for listening to this week's episode of Transmission Control.